Rory to introduce today's speaker.
Yeah, it's my pleasure to introduce Dr. Susan McRae, who many of you know is a sabbatical visitor here in the MVZ. So before I forget, I'm going to tell you where she is, because she's interested in interacting with as many of you as possible. So she's in the lab past my office, so right at the back end of this part of the MVZ, the nice, quiet part, I would say the happy part, the extra happy part. Anyway, just a little bit of background on Sue. She completed her undergrad degree at Carleton University, and then completed a Master's of Science degree at the University of Waterloo, and then spent quite a bit of time at Cambridge in the UK, where she did both her PhD and her first postdoc before sort of jumping continents and moving to Strate, where she did a second postdoc. And she's currently a tenured professor at East Carolina University. She does a lot of very diverse stuff, and she's going to highlight a little bit of some of the different aspects she's interested in today, but is mostly going to talk about this remarkable system, this ruff sandpiper, which just in the past year there were 2 really influential papers published in Nature Communications, of which she was an author of one. There's an interesting story behind the twin papers. But it also got highlighted in last week's Science magazine, for any of you who read on these super genes that are being discovered that underlie a lot of bird phenotypes. So I'll end off my introduction with one interesting fun fact, and that is that Sue once helped rescue a zebra, or a zebra as you guys would say, from drowning in a swimming pool. Thank you.
But I probably won't be talking about that today. And so thank you very much, Michael, for hosting me, and Rory and everyone. It's been a pleasure to get to know a lot of students already here. I'm really looking forward to getting to know more of you as well. And as Rory pointed out, I wanted to begin by introducing myself a little bit, because although several of the other speakers that we've seen here recently have had affiliations with Berkeley, I haven't before been affiliated with Berkeley, so I thought I would tell you a little bit about my background, where I came from, just by giving you a few research highlights. So I was an undergraduate at Carleton University, and I worked on parental care in American robins with Pat Weatherhead and Bob Montgomery. We were working at the Queen's Biology Station in Ontario, and I was interested in pre- and post-fledging care in robins, and particularly worked on sibling competition, and this was some of the first research to look at the influence of sibling competition on distribution of food at the nest, and in a species that was non-siblicidal. Okay, and also some of our work on the parental division of labor, on care in overlapping broods, developed some theory that about basically how extra-pair fertilizations might be expected to be distributed across broods in multi-brooded species. For my master's, I then thought that I should probably be working on a Canadian animal, not an American animal. So I started working on the hooded seal, and we looked at mating strategies, including the paternity, basically, of these animals using what was a newly minted technique back then of DNA fingerprinting. And so mainly I focused on the precopulatory mate guarding of the male hooded seals. But then I came back to my senses and began working on birds again when I went to Cambridge to study moorhens. I was working with Nick Davis and focusing on conspecific brood parasitism, where females lay some of their eggs in the nests of neighbors. So these are laid by 2 different females here. And also joint nesting, where females lay in a collaborative setting. So they basically have 2 females cooperating with a single mate, laying in the same nest. And so one of the things I was interested in was how these 2 strategies working in the same population, you know, the interplay between parasitic and cooperative strategies in facilitating tolerance, really, of another female's eggs in one's nest. and a bunch of field experiments and so on. Later on, I was a postdoc at Cambridge, actually focusing on weaver bird cooperative breeding, but I was distracted and went off to Bushman Land, Namibia 1 year with Ian Jamieson, and looked at similar brood parasitic strategies among lesser moorhens and red-knobbed coots. They also have conspecific brood parasitism, and they're very, very good at spotting parasitic eggs in their nests, and both species bury them in their nests under the rest of the clutch, so that's quite clever. So that was a really fascinating diversion. And then for my postdoc at the Smithsonian, I went back again to working on common moorhens, called common gallinules around here, I think, and looked at brood parasitism in tropical setting, and also studied purple gallinules, American purple gallinules in parallel, which are non-parasitic. So the moorhens have an intermediate level of parasitism between these African and British species, and then the purple gallinule has no parasitism. So I was sort of comparing the different populations, and I would have loved to have gone around the world continuing studying moorhens, but I couldn't because I had to get a job. And I'm skipping over a few years here, but most recently I've been working on this fellow Kingrail with my graduate students at East Carolina University, and we work on them way on the other coast of America. So we're in North Carolina, right on the coast there, and we've developed genetic markers. We've been looking at population genetics. We've also been tracking them, looking at home ranges, movements. One of my students was interested in climate change variables and how that affects nesting success. We've started working on communication now. So I'll probably be speaking to Steve Bessinger's group about this system a little later if anybody's interested. But right now, I'm going to talk to you about these wonderful rough sandpipers. And so this is a project that my co-author, Doug Lang, has been working on for more than 30 years. I was fortunate to be invited to join him when I was on sabbatical 10 years ago in Simon Fraser University. So this has been kind of a 10-year odyssey for me. And basically my part of this project was to help Dove work out the behavioral repertoire of the female mimic morph of the rough sandpiper. And so I'm mostly going to talk about the behavioral repertoire and how we came to work that out. But I'll also touch upon the papers that Rory mentioned in the introduction. And I'm not going to focus so much on the genomic aspects, but how the discovery came about. I'm going to give you the backstory to how this discovery came about. So, permanent genetic alternative mating strategies. We heard a little bit last week about Barry Sinervo's Uta lizard system, where you have blue-chinned males that defend small territories to attract females for the purpose of mating, but they can be cuckolded by the orange superterritorial males that have larger territories that encompass the territories of blue males, and they in turn are cuckolded by yellow-chinned sneakers that are moving around and sort of parasitizing opportunities for matings within the territorial system. So they have permanent genetic male morphs. A very different system is that of the sponge isopods that are studied by Schuster and Wade, where you have alpha, beta, and gamma males. The large alphas are territorial, defending harems of females within the sponge. The medium-sized beta males mimic females to obtain matings, and the sneakers are much smaller, the gamma males that sneak around and go unnoticed as they seek reproductive opportunities within that system. There's a fish that has been described, a swordtail where small males chase and large males court. And then there's the ruff. So the number of species for which permanent genetic alternative strategies have been described are fairly small, but you can see that they encompass a wide variety of different animals. And the ruff is very, it's kind of iconic as a model for sexual selection with its wonderful rough display feathers. The species name, Philomachus pugnax, actually means pugnacious male lover. And Darwin, in his The Descent of Man and Selection in Relation to Sex, mentioned the ruff no less than 10 times. But the ruff is actually very different from the other birds in this order. So order Charadriiformes, family Scolopacidae, the shorebirds. They're not always coastal. These are some North American representatives. Some of them live upland. These smaller ones are all in the Calidris clade and may be familiar to some of you birders. Birders tend to call these smaller Calidris species peeps. Particularly when they're migrating in the winter in the dull plumage, they are rather difficult to distinguish from one another. So again, they're sort of called peeps just generally. And it turns out that phylogenetically, the ruff is placed nested within the Calidris clade. So this is from a super tree that was published in 2004, and these are all Calidris. Some people changed the genus name of ruff to Calidris as well. But they're very, very different from other Calidris species. They are not monomorphic, they are not monogamous, they are not monotone. They are an old world sandpiper, and they overwinter in mostly Africa, and then on their migration on the way to Eurasia, the males molt into an ornamental plumage, which is quite striking. They use their ornamental plumage to display on the lek. Lek is a Swedish word for games without rules. And so this is a sort of a free-for-all. They're displaying and trying to obtain matings on the lek. Again, they use their sexual dimorphism to full effect, showing off their feathers. And like most lekking species, some males get lots of matings and then other males get very few. And all the females get on the lek is a mating, because after the mating is over, the females go away and nest by themselves and raise the chicks alone. So they have uniparental female care. One of the most fascinating things about the ruffs is this amazing polymorphism. The ruffs in general are very variable in their ruff coloration, and the plumage polymorphism is associated with a behavioral polymorphism. We have two types of males generally shown here, with the dark plumages being the independents, and the ones with very striking white plumage on their ruffs being called satellites. And so just to orient you with these male morphs, the independents have dark ruffs and or head tufts, and they behave on the lek as residents. This is a territorial behavior where they exclude other independents from their court. 65% of the males, the total males, are independents that are unsettled or non-breeders in any given year. So only a proportion of the residents actually behave territorially, a proportion of the independents only behave as residents in any given year. Satellites, on the other hand, have these white ruffs or white and golden tufts. They represent 15% of the population in any place in Eurasia where you go and study them. They're approximately 15%. And they are non-territorial, moving among the courts that are being defended by the residents and co-displaying with the residents. And so theory predicts when you have permanent genetic mating strategies that you should have approximately equivalent fitness between the different alternative strategies. And just as in Barry-Cinervo's lizards and Schuster and Wade's sponge isopods, the ruff shows balanced, basically the mean annual reproductive success per male is not significantly different among the morphs. So you basically have similar life history strategies between the independent and the satellite morphs. So how is the polymorphism stabilized? Basically, females prefer to mate on courts that are that have satellites on them. So they are attracted to these courts with the satellites on them. They probably are flying in and seeing the white plumage, and that seems to attract them. And so it behooves males to cooperate in these situations where satellites are co-displaying with the residents. So my co-author, Dov Lank, at Simon Fraser University, he imported eggs from Finland 30 years ago, and he established a captive colony of ruffs in order to study the genetic basis of this polymorphism. Beginning with about 31 founders, he established this breeding colony that's been going ever since. It's currently housed in Simon Fraser University at the top of Burnaby Mountain in the Greater Vancouver region. And the ruffs actually take very well to captivity. They form leks in captivity. Their courts in nature are only about 1 to 2 meters squared, and so they don't actually need much space to do their thing. What you see here is an independent behaving as a resident. He's got 2 satellites behaving with him. These are females. You can see the females here are not that interested at this point, but they're doing their best to attract the attention of females who may be interested in copulating. And you can see that they're using their rough feathers to display. A lot of what they do is posing. This is male on male mounting, and you could see that the females suddenly got a little bit interested. So that's probably the function of that behavior. When they mount one another, they're just trying to incite some interest in the females. They do that quite a lot. Now, in this next frame, you'll see what the purpose of all the displaying is. So here's a female that is interested. She's soliciting, and she crouches, and you can see the resident is mating with her there successfully. And basically, that's kind of what this is all about. So here's another female interested. But if you'll notice, there's one of the satellites has noticed that the female is interested and is trying to surreptitiously come in there. But he gets dominated by the more aggressive resident. He doesn't actually get to mate, but he has succeeded in interfering with the mating of the resident. So, you know, when females do present themselves, there can be conflict between the co-displaying males over who gets to mate. So, using a pedigree approach, Dov Lank and his colleagues were able, quite some time ago now, 1995, to work out that the satellite morph behaves, it's inherited in a Mendelian fashion, and it behaves as a single locus with 2 alleles, with the satellite allele dominant. And this is based on a model which is an autosomal trait, and because it's autosomal, he surmised that, well, we should be able to produce this behavior in females as well. And indeed, if you implant females with testosterone, you can induce male behavior in females, and that happens after a couple of days after implantation. But then about 6 weeks later, the females that are implanted also begin to grow ruff feathers. And so that's the way that they were able to type the females as either independents or satellites. And when you incorporate the female type into the model, the genetic model based on a single locus with 2 alleles still worked. So at that point, that was all there is to know about this fascinating ruff system, right? Wrong. Turned out that there was another morph of male that had gone unnoticed in spite of about 6 or 7 decades worth of research by multiple research groups, mostly Scandinavian groups, on this species. And this paper described this new morph, so permanent female mimics. And one of the interesting things about how the mimic was found is that the lead author, Yukuma, is not a scientist but a potato farmer. This is a wonderful case of citizen science. So Yukuma enjoyed weekend banding with his Dutch Wilsternetters. This is just a group of the members of the general public that enjoyed birds, loved to go and band shorebirds on the coast as they were migrating through in Holland. And Tunis Piersma is a professor at Groningen University in Holland. And so the two of them were looking at the records of these banding records of ruffs returning on their fall migration. And they determined that there was an intermediate size bird that they determined to be male based on, they actually killed a couple, opened them up, found testes. And they called them faders, which is it's the old Dutch word for the fathers, based on the fact that they thought that maybe these were the ancestral type of male, and also they actually proposed at one point that they might have parental care like other sandpipers. But anyway, they describe these males as being small and having female plumage. This is a plot from the paper, this is a figure out of that original paper, showing that the faders have a measure of body size, wing length here. They have discrete and intermediate size distribution between ornamented males and females. And this is just to show there's only 22 birds in this distribution. This is scaled so that it's only a very, very small proportion of the males that are in this intermediate size category, less than 1%. So this is based on 2,540 males that were banded. So we have a third morph of male in this system. So what do we do with them? So the faders are female-like. They have female-like plumage with no ruff or head tuft. They represent a tiny proportion of the population, and nobody has seen them behave on a lek. They go unnoticed. And so this is where I was fortunate to come in. So Tunis Piersma captured 2 faders on migration and kept them for a year. He had intended to try to breed them on his own, but he didn't have a colony set up. And so he had a lot of difficulty getting them to breed. And so he offered them to Dove Lank to see if Dove could get them to breed. Dove has a green thumb for breeding ruffs. And I happened to arrive at the same time as the faders did in Vancouver, which is how I came to work on them, because Dove invited me to help observe them. And basically, again, there were no observations of mating by these guys in the wild. We knew from Eubuchma's descriptions that they had mighty testes, and we thought, well, perhaps they're going to behave as sneakers or maybe female mimics. And so we set up breeding design. We put the 2 faders in an aviary with most of the females that were in the population in the colony at the time, 61 females. And then in an adjoining space, we put 2 independent males and a satellite, separated just by a thin netting so they could see one another full time. Now the purpose of this was we really wanted to breed the faders because one of the main things we were interested in is seeing how, if and how the morph was inherited. So we wanted to breed them as much as possible. So this gave them the best possibility of having access to females, but also being able to observe their behavior with the males. And every morning we removed the netting and allowed the males to interact with one another in order to observe how the faders interact on the lek. So we just did that for about 2 hours every morning, and the males would, the faders would join in with the males when they were lekking. And then we actually rotated the ornamented males every few days so that we kept them fresh. And just basically, what we found was that the faders did not display, but they did stand close to the most actively displaying males. They were not chased away. They were courted by the males quite commonly, sort of like they court females. And the faders also crouched and were mounted by the ornamented males. But you saw already that males do that to one another quite a lot anyway, so that wasn't very unusual. In fact, this photograph is of a fader on a Norwegian lek. So now the Dutch Wilster netters put lots of color markings on all the faders that they catch so that they can be spotted on leks. And this one was spotted on a Norwegian lek, but they saw him mounted by males, but they never saw him mate. So they've still never, as far as I know, nobody's ever seen a fader mate in the wild so far. But we were able in captivity to describe their repertoire on the lek, and it's very different from the other males. So just briefly, these are means based on individual males and then standardized across the population in the experimental trials. And so what you can see is that the resident males are much more aggressive than the other morphs. They display a lot more. They tend to stay away from other resident males, so they have a lower score for proximity. But they're not very alert. And, a lot of times when they're displaying, males miss things. They actually don't notice when females are soliciting them sometimes because they're so into their display. And so the satellites, by contrast, are less aggressive. They display a lot, but they always are close to a resident when they're displaying. And they're a little bit more alert. A lot of their postures are a little, with the head raised a little bit more. They do an oblique posture quite a lot. By contrast, the faders, they are not aggressive. They don't display. And they do stand near the actively displaying males. And they remain alert, and that is the secret to their success. And so they do spot the females that are soliciting when they come by because they're not doing the display. And so this is, I'm going to show you a video of the 2 faders. This is one, this is the other, standing close to some actively displaying males. And you just keep your eye on the blue fader. Let's see if I can stop this at a key time. Let's start it. Okay. So here's one fader, here's the other. We've got a resident and a satellite, and they're doing a co-display. And again, the resident's got head down, it's not really noticing things too much. This is still the blue fader here. He was the more active of the two faders that we had. She had quite different personalities. Okay, whoops. Oh, okay, sorry about that. All right, just start this again. I just wanted to stop it at one point to orient you. Okay, so when the frame changes again, I want you to look up in this corner because a female is going to come in and crouch, and I want you to watch what the fader does when she comes in and crouches. So he's going to be up on the brick in a minute. There he is. So there's the blue fader. Just watch up in that corner. He tries to get in between the resident and the female, and watch him again. This is the fader. When the resident comes back, he kind of fakes him out. He does a little crouch. Now, that wasn't the best video, it was handheld video, but we used to see that quite a lot, that they decoy the ornamented males away when the females show interest. And so, fader mating tactics, we saw both of the faders sneak, so they were mounting females that crouched for displaying males. They were also chosen by some females when, for example, the other males weren't in the pen. We saw females crouch right in front of the fader, so they're recognized as males for sure by the females. And one of the two faders particularly did a lot of this mimicry behavior where he crouched to decoy the ornamented males away from soliciting females. So they are behaving like sneakers and mimics. At the same time we were doing these observations, Tana Morgan was working on her master's thesis, and she was interested in hormone levels among the different morphs. And these are some data from her thesis. They're also in the Nature Genetics paper, sorry. What you can see here is in the peak of the breeding period, androstenedione is, which is a precursor to testosterone. It's on the biosynthesis pathway to testosterone. It's very elevated in both the satellites and the faders, but not so in the independents. Whereas in the independents, testosterone peaks in the main part of the breeding period, but not in the other two morphs, the satellites and the faders. So there are differences in the profiles, particularly between the independents and the other two morphs. I'm going to come back to that in a bit. But just to show you that first year, the faders had many more successful mating attempts, and all attempts at successful matings and all mating attempts were much higher by the faders in our design. Now I'll just point out that we used a design that stacked the deck in favor of faders because that's what we wanted. But it is remarkable how successful they really were. They were chosen, or were effectively mating with 15 different females. These are of the ones we reared. So we actually started with over 200 eggs that season, and we only successfully reared 41. That's not unusual. These are not fruit flies. They're difficult to breed. And so we get those kinds of rates of success. But most of the chicks reared from that pen that year were fader offspring. And two of the females actually had offspring from both faders. So they were quite popular at that stage. Now, again, looking at the genotypes genotypes at this point, but looking at the paternity of the offspring here, we realized immediately that some of the fader sons were not fader morph themselves. And right away, we knew from that this was not, for example, a homozygote version of the satellite allele. So we were able to make some eliminations along the way as we were trying to figure out the inheritance system of this new morph. We then set about building up the pedigree. So we developed a pedigree using parentage from microsatellite data. In all, we constructed a pedigree of 64 families going back 10 years before the introduction of the faders and 4 years beyond it. And this was part of Lindsay Farrell's PhD. So she developed new microsatellite markers and also mapped, using CRY-MAP, she mapped the morphs onto the pedigree, did a linkage analysis. She, again, developed 43 new microsatellites, and then she blasted all 58 microsats that we had against both the chicken and the zebrafinch genomes to establish that the markers were well distributed around the avian genome. And she also made an interesting discovery on chromosome 11. And so basically, the main results from her thesis were that the fader morph assorted as a single autosomal locus with the fader allele dominant, very like what we found, or what Doug found with the satellites before. And also that there was co-segregation of one of her microsatellites on the chromosome 11, and it actually was very proximate to the melanocortin-1 receptor gene. And so that was a very big breakthrough. We now had at least a chromosomal position. But to go forward in deciphering the genetic architecture, Doug worked with Taylor to get some funding from the BBSRC and to do a full rough genome. And that's when the genomic work began. So it was, they hired Clemens Kucker as a postdoc on the project. He was the one who began the genomics, and so they did first rough genome, reference genome of an independent male. They then also did transcriptomes of 5 other roughs from the colony of the 3 different morphs. And in addition to that, high-density RAD sequencing of some unrelated individuals, some from Dutch population, also from a Finnish population. And again, I'm not going to go into a lot of detail on the papers that were published last November, but to make a long story short, found a supergene. And this is a 4.5 megabase chromosomal inversion. So the new linkage map of chromosome 11 based on the genomic data showed that there's a 70% smaller linkage map when you include the independents only compared to when you use all individuals. That's how they recognized it was a big inversion in this chromosome. These show the probabilities of association among the SNPs using the entire rough genome aligned based on the chicken genome there. And you can see from this, these are blow-ups of this region here, showing the fader in relation to independent and then the satellite in relation to independent. And this is the location of the chromosomal inversion. So there are 2 different variants. There's a satellite variant and there's a fader variant of this chromosomal inversion. Now, a second research group also published in the same issue of Nature Genetics and found at the same time, using a completely different genome strategy, they found the same chromosomal inversion. And so the two groups agreed to publish the papers at the same time. And basically, these are some figures from their paper. In addition to showing the inverted regions here, they focused on the fact that the satellite and fader version are virtual. There's kind of a mirror image pattern between them. And they use this to determine that they believed the original inversion was the fader version, so that was an inversion that might have occurred about 4 million years ago, and then a subsequent inversion of this part called region B in blue here occurred maybe 500,000 years ago. And so that is, you know, possible order of how these two different variants came about. They also described some accumulation of non-synonymous mutations in the inverted region, and particularly they focused on some deletions that they had noticed, which will become significant in a few minutes. But I want to just mention the significance of some of the genes that were identified in the annotation. So these are genes that were found to be associated with the polymorphism and with the chromosomal inversion. And obviously, the difference between satellite and resident plumage is very striking. The white plumage of the satellites is most likely to be related to the melanocortin receptor gene difference that was found within the inversion. Some of the other proteins that were associated are known to be expressed in gonads, so that might have something to do with the differences in testis size among the different morphs. Both the satellites and the faders have larger testes. And the steroid metabolism genes that were found, again, both groups identified these genes as being important in the inversion. In fact, this first one is associated with those deletions that were identified by the Lama-Cheney group. And those most probably are what are responsible for the very big differences in aggression between the different morphs. So there seems to be kind of a knockout effect of the level of testosterone there. Also, our group identified steroid receptor gene regulation difference in the satellite and fader inversion. And if you recall, when testosterone is implanted into females, we can induce the behavior in male behavior in females and growth of a ruff in females. So we were curious about what happens to faders if you implant them with testosterone. And actually, Tana Morgan did this experiment back in 2006. And what happens is not a lot. They don't display, nor do they grow a ruff. So there's probably some disruption to their receptor gene as well. And I wanted to show you this one more video here. This is footage of the females after they have been implanted, and you can see some of them are behaving like residents. So they're being really aggressive and running around. And this one that's crouching here, that's a fader. And so this one fader, who was the one that didn't decoy a lot before, got really randy when he saw the females behaving like males, and he started behaving like a female. So that was kind of interesting, but a little bit anecdotal. And there he goes again. You know, he got really excited, and But they didn't display, so that the important thing is that they didn't start becoming aggressive themselves. So I think that has something to do with the lack, or maybe some sort of knocking out of the receptor gene. Another gene that was found to be significant with the inversion by both groups is the centromere protein N gene. So this is a critical gene involved in centromere assembly during mitosis, and it was broken by the inversion, and so it is not functional in the inverted version of the supergene, or the superallele. I want to actually introduce the word superallele and sort of market test that on this group, because I think it better describes what these are, these two variants, but we can talk about that later. So the fact that this critical gene is in the breakpoint of the inversion suggests that having homozygosity for the inversion would be lethal. And indeed, we found evidence for this based on individuals, the proportions of individuals in the population that were faders and satellites. And we found that basically there's a deficit of homozygotes for both the fader and the satellite variants. And so that appears to be lethal. We don't find those. And that also, by the way, goes some ways to explaining why we had some difficulties with our breeding experiments. After that first year, 2007, 2008, we had we were trying to back cross the fader daughters with the other fader. And we had virtually no success in doing that. We could not get the fader daughters to breed, and they weren't laying, and they then when they finally did lay, we couldn't raise any of the eggs. So that is probably why, because there is this lethality of homozygotes for those two variants. So another aspect to the phenotype is that, of this polymorphism, is that the independents are significantly larger. The satellite males are sort of intermediate in size. They're just a little bit smaller on average than the independents, but the faders are very much smaller. And this is to their advantage because it allows them to appear more like females. But the females also inherit this characteristic. And so this is something we published a little bit earlier, was that once we had started to breed the faders into the population, this separate discrete category of females appeared. So these are offspring bred since the faders were introduced into the colony, and these are independent size ranges. They're all, the body size is a principal component, composite measure of body size, and it's all standardized. So the residents are the largest, the satellites intermediate, the faders, fader males form a discrete population, and then the females have this bimodal distribution, and we assume that these are the females carrying the fader allele. So again, none of these females have bred successfully. So what we're thinking is there might be sexually antagonistic selection on this allele, the super allele, which causes male carriers to be very successful in their reproduction, but has a deficit to female reproduction. Now, all of this may seem somewhat familiar to you if you are cognizant of this other example. There is a very common species that's not found in these parts. It's found in Berkeley, but in the east, the white-throated sparrow is an extremely common bird. And we now know that it has a super gene. In fact, the pericentric inversion in avian chromosome 2 was first published in 1966. Because it's a pericentric inversion, it's very easy to spot in a karyotype. And so it was actually cytological work on this bird that first identified that particular inversion. And Thornycroft also realized at the time that it perfectly coincided, the white morph with white plumage, white stripes, coincided perfectly with the possession of the inverted variant of that chromosome. So this is very familiar, a plumage polymorphism in so producing two different variants coinciding with a chromosomal inversion. There's 50% of the birds in the population have the variant. And it turns out they mate disassortatively. So in the white-throated sparrow, white males always mate with tan females and the reverse. And again, there's a lot of similarities. There's two morphs in this case in both sexes. The heterozygotes are the white striped ones, the homozygotes are the tan striped ones. The heterozygotes have high aggression levels. The males sing more, they're more promiscuous, they have high sensitivity to steroids, and in fact a recent paper found a fixed difference in the inversion variants that regulates the transcription efficiency of the estrogen receptor 1 gene, which codes for a receptor that's found in the brain. And a recent field study showed that local expression of that gene is linked to the genotype. So they know quite well that the behavior is linked to this genetic variation. Now, a recent paper that I want to plug that also came out this month talks about the origin of the inversion in the white-throated sparrow. It's a little different from the ruff, but I want to plug it because three of the authors are at my home institution, East Carolina University. So Michael Brewer, formerly of Berkeley, did his postdoc here and then came to us in a faculty position. My colleague Chris Balakrishnan and his student Daniel Newhouse all are on this paper. And they basically describe how the inversion is maintained by disassortative mating. They describe how the homozygotes, again for the variant, are also believed to be lethal, at least in males. Very few homozygote females have been described, but they are very aggressive, and the males don't seem to survive past juvenile stages, but again, there are very rarely reported female homozygotes, and they're extremely aggressive. And so one of the things that they found in this paper was that there seems to be a very ancient origin of this white superallele. And so they used full genomes of a variety of sparrows that are now available, and constructed a phylogenetic tree based on the full genomes, and then separately on the inverted region of chromosome 2. And they found an ancient origin that precedes the branching of white-throated sparrow from its sister species. And so they inferred that probably the existence of the two variants in the white-throated sparrow population arose through interspecies hybridization. So, basically, that introgression of the white variant into the population at some later stage. So that's kind of cool and very different from the ruff. But the other thing they describe here is evidence for functional degradation of genes within the superallele. And again, this is kind of what the Lamicheney group was also describing. So the accumulation of non-synonymous mutations and then followed by some deletions. And that all is very reminiscent of what happens in sex chromosomes. So it compels us to think about maybe these supergenes are actually nascent sex chromosomes. So in typical chromosomal sex determination, whether it's an XY system or a ZW system with male or female heterogamy, respectively, the Y and the W chromosomes are famously degenerated. The portions that no longer are recombining with their partner chromosome become degenerate and degrade. There are now quite a few species that have been described, a handful in mammals and quite a few in fish as well, that have polygenic sex determination systems. And in some cases, they're a single locus, or in other words, basically a second kind of chromosome analogous to the Y arises and can be interchanged, and this produces strange sex ratios. In the case of this mouse species, the W overrides the Y and reverts that mouse to a female genotype. And also multilocus polygenic sex determination, which has been found now in quite a few fish species and is studied by Reed Roberts at North Carolina State University. And so again, these polygenic systems produce odd sex ratios. In this case, he studies a cichlid fish where the new variant produces this orange blotchy morph that is advantageous to females because it gives them crypsis, but it's disadvantageous when it occurs in males because it disrupts their breeding coloration that they use in display. And again, it produces very odd sex ratios. So this may be something that many other systems should be looking for. One of the things that this review paper mentions is that these kinds of systems can be missed if you're sampling in the field and you're taking small samples. You might actually fail to collect some of the key individuals that would allow you to understand, the full gamut of genetic variability in the population. So this is just an idea that we've been discussing and kicking around, that these superalleles that we're finding in ruffs and white-throated sparrows are basically new sex chromosomes that are just in the very early stages. So, bringing us back to the ruff, what does all of this mean? So, faders, which is the main topic of my talk, they are in fact avian female mimics. They do represent a third permanent genetic morph. They are controlled by the same, a variant of the same supergene as the satellite. The chromosomal inversion maintains the male polymorphism system in the ruff. Faders themselves have a different behavioral repertoire from the other males. They don't display, but attend displaying males on the lek, and they attempt to mate with females that approach. And the cryptic female plumage and small size that are part of the phenotype of the fader facilitate sneaking behavior and increased tolerance by the resident males. And it leads us to think about some of the future directions that this research could take. One of the things that we should revisit is characterizing the relative fitnesses of the different morphs, particularly looking at morphs of both sexes, because it offers the opportunity to study sexually antagonistic selection and strategies that females might use to mitigate the effects on their fitness. For example, when they mate with faders or with satellites, they might want to skew the sex ratio in favor of males. That would be to their advantage. And we also have a lot of work to do now elucidating the functional differences in the morph phenotypes. We have some candidate genes now. There's lots of opportunities to look at their effects on the morph, possibly with some gene editing approaches. And it also, I think, both of these systems compel us to reconsider how we think about sexual identity and mate choice, and particularly strategies of individuals, given that they're not necessarily able to mate with all of the individuals of the opposite sex within the population. It really constrains the numbers of possible mates available to individuals more than we thought before. For example, in the white-throated sparrow, only a quarter of the individuals in the population are available as mates when you think about it. So just before I go, I thought because we're sitting in a library that I should point out my co-author discovered quite recently this descriptive note by a fellow called Stoner, unfortunate name, but he described a single case of a male ruff in the plumage of an adult female back in 1937. And so these were brought into a British zoological garden, and he recognized this individual as being a very small male, or at least an intermediate sized individual. It had the misfortune of dying, and so he opened it up and he discovered that it had very large testes. And this is actually a figure from the publication, which I think is great. Wouldn't it be nice if we could just like hand draw? That was about that size. And so these are, by comparison to this female mimic, a male in another male in breeding condition and a male in non-breeding condition. So he had the size, he had the testis size. He describes within the paper some observations made by another person in 1907, where he saw females mating with one another, which may have been actually female mimics. And so maybe this isn't so new after all, but I think it emphasizes that we should make use of this dusty literature, and because there are treasures to be found there. So I would like to thank all of our co-authors, the students that worked on the project, Tunis Piersma for providing the ruffs and the opportunity in the first place, and certainly my co-author, Doug Lank, without whom I wouldn't be talking about these today. So thank you very much for listening.
If somebody could get the lights, maybe we'll let people who need to sneak out do so, and I'm sure Sue would be happy to take questions.
So do you know if they're going to try any of this new CRISPR technology? Is Doug thinking about doing it?
Well, Daniel Newhouse, who is a CRISPR Christian student, has just taken a course on it. So it's not completely outside the realm of possibility. But at this point in time, I'm not aware of anybody who has successfully done CRISPR on birds. It's going to be really challenging, given that they are inside of hard-shelled eggs. But it is something that we have talked about a little bit in the future, far in the future.
So I was curious about this sort of analogy to sex chromosome systems. So when sex chromosomes evolve, you get degeneration of one of the two chromosomes, not of both.
Right.
Because of suppressed recombination and one being present in two copies in one sex. But so in your case with these inversions, where is the degeneration seen? Is it in one of the alleles?
It's only in Yeah, it was only in the satellite and the fader allele, the inversion alleles, and it's only in the inverted region. Now, to be fair, I guess, I don't know if it's been adequately looked for in other regions of the chromosome, but it is within the inversion that they noticed high numbers of non-synonymous substitutions. And in both the ruffs and the white-throated sparrows now. And I guess the idea is that functional degradation should occur first, and once you've got functional degradation, then you'll get deletions occurring subsequently. I think that's probably the order of you know.
But it's in just one of the alleles.
In just one of the alleles, yes. And of course, the fader and the satellite allele never occur together because they would also be lethal. So,
But
so
no frame shift mutations yet. How many genes are in that?
So it's different in the white-throated sparrow and in the ruff. There's about 100 genes estimated in the ruff inversion and about 1,000 in the white-throated sparrow. So they're quite different in size. But you know, kind of one of the interesting things is that the genes that are within these inversions are similarly characterizing things like plumage and hormones and receptors and things like that. And so it may be that inversions happen in other parts of the genome and don't have these kinds of effects, but when they happen to hit upon these kinds of genes crucial to fitness, sexual selection, and so on, that's
Yeah.
what it produces.
That's fascinating stuff.
So when you implant the females with testosterone, they run around and act like independent males. Or have you tried implanting fader females, like females with the fader gene with testosterone? Would they behave? Because the faders they wouldn't chase everybody around, right?
To my knowledge, no one's done that yet. But it's possible they've done it, you know, when I wasn't there and I don't know about it. But to my knowledge, they haven't done that.
Is that
but it's an obvious
behavior difference
experiment
is really
to do.
interesting, that the faders have to know to not, I mean, they have to behave really differently from your
Right, and I think that's part of the linkage. I think the fact that the plumage genes and the genes involved in steroid metabolism are in the same region, I think it's very crucial that all that is together for this system to work. So I mean, I don't think that faders have to know anything. They're just, you know, they can just behave mechanically, if you like. I mean, they're you know, it just, it's evolved independent of any sort of conscious thought.
But I'd just be interested to see what happens.
Yeah, no, it's definitely something that we should do.
Cool. Well, maybe given the late hour, we'll leave it there. And if people have other questions, you can come up and ask Sue. Thank you very much.